Good evening, everyone. It is 7:04, 1904 for those of you on military or circadian time. Today, Brooke Morren is going to be presenting about retinal degeneration, specifically AAV, which she'll tell us what that is, TXNIP, which we've been calling TXNIP, prolongs cone survival and vision in mouse models of retinitis pigmentosa, and that is Yunlu Zhu, I think is how it's pronounced. And that is in the SEPCO lab at a place called Harvard Medical School. And that is published in eLife. I think Brooke is going to mix it up a little bit because she feels inspired. So go ahead.
Okay, let me first present it. So I'm presenting how AAV TXNIP prolongs cone survival and vision in mouse modules of Retinas pigmentosa. So AAV is adeno-associated virus. Next slide. So Retinas pigmentosa is an inherited genetic eye disease where there's an initial degeneration of rod photoreceptors which affect night vision, followed by a loss of daylight vision onset by a damaged cone photoreceptor. It is believed that Retinas pigmentosa was caused by a shortage of glucose in cones, causing them to starve and die. And 20 genes were tested to improve vision, but only TXNIP had a beneficial impact. So, next slide. Okay, so here we see how glucose enters the photoreceptor and undergoes glycolysis, in which it becomes pyruvate. The pyruvate can either oxidize if there is oxygen, but if there is no oxygen, the pyruvate undergoes lactic acid fermentation. And so if you look to the photo on the left, in the center, which is highlighted by the purple box, this shows glycolysis in the center, and it's actually in Dr. Char's room. So TXNIP's mode of action has at least two pathways. One of the pathways is to improve the mitochondrial health, and the other is through lactic catabolism. So to better comprehend, we can use the central analogy of muscles. And if you don't work out and then suddenly you do, you get sore due to the buildup of lactic acid. Under these circumstances, rod and cone photoreceptors do not want lactic acid because it will ultimately kill them. So TXNIP is inhibiting glycolysis, which entails that there's not as much lactic acid buildup. So similarly, TXNIP is specifically increasing and activating the catabolization of lactic acid, and the presence of oxygen then leads to aerobic respiration, in which the pyruvate goes into the mitochondria and begins oxidation. So this process produces ATP, and retinal cells are not healthy if they undergo anaerobic respiration, and they are ultimately desperately trying to survive. Next slide. So this was figure 1, and it was labeled as TXNIP effects on cone survival and cone-mediated vision in RP mice. So P20 stands for 20 days postnatal, and P20 is the day the mice are weaned from their mom. Around P50, vision is already getting lost in the mice. So in figure A, this shows the retinal scans, which are stained to show the AAV-encoded TXNIP and H2BGFP. And B, as shown in figure B, P20, there was no significant difference when using TXNIP. By P50, there was a significant 60% increase in using TXNIP. At P130, TXNIP is still working, and there's still a significant difference when TXNIP is present versus the control. So if we look at C, this figure shows how mice and activated TXNIP have a greater success rate of having vision in comparison to the RD10 mice, who do not have TIC-SNP activated. And this shows there's a detectable and positive effect when using TIC-SNP. And the other part of C displays direct comparisons to the control of mice and mice who have activated TIC-SNP. So then, as we go to the next figure, which
Not yet.
is...
I just want to make a note. This thing right here, the ability to do this figure right here, C, and then the one on the right as part of C, which is weird that they just kept it C. This is something that you will likely have to replicate in your first year of biology in college. This is a classic comparison. It's a good way of, they did this well, and so I think that's worthwhile to note. Okay.
So for this next figure, figure 5, it's called the effects of TIC-SNP on RP cone mitochondrial size and function. And Dr. Char wants me to ask if anyone would like to explain part A of this figure.
Brooke would really like to ask someone, not Dr. Char. Brooke would like to.
Oh, sorry. Brooke would like to ask someone.
So we got the P20 RD1 on the y-axis, and on the x-axis we have the control, the TXNP C247S, TXNP S308A, and then
That's good.
okay, thank you.
Yeah. So what is it?
I have no idea.
So basically part A of figure 5 is showing the scans of mitochondria and how TIC-SNP and C-SNP had an improved mitochondria in comparison to the control. So if we look at the initial image, the control does not have as strong of a mitochondria as when TIC-SNP is used or incorporated, as well as TIC-SNP C247S.
And just to be a little more specific, what you mean is this looks like crap, right?
Well, I wasn't going to say that.
Yeah, it's a very unhealthy mitochondria, whereas this is a pretty healthy, and this is a really healthy mitochondria. And then this one, it kind of looks like crap again.
Yeah.
Yeah.
What about it is indicative of it, like, not being healthy? Like the shape or whatever?
Well, mitochondria tend to have the classical shape. Tend to. This is not always true, but they tend to have the classical shape you see in textbooks. So they're either kind of like oblong or they're very round. I've noticed this is a transmission electron micrograph, so this is zoomed in something like 100,000 times, probably more than that, almost 1 million. And I've noticed they tend to be more roundish, but when you see them really start to bleb like, well, let me click it again. When they start to bleb off like this, it's almost like, imagine that these are cells, right? And when a cell goes through apoptosis, this is kind of what it looks like. This is not a healthy cell. And then you can actually test it using certain stains to see if the mitochondria are healthy or not, which they are not here. And they are not so much here, but they are here. Make sense? Cool.
Okay, so then we'll go to the next figure, which is figure 8 in the paper. And this was called the effects of combinations of TIC-SNP C247S with BEST1 NRF2 or TGF beta 1 on RP cone survival. So BEST1 is a promoter for genes only in retinal pigment. And NRF2 is a gene with an antioxidant damage and anti-inflammatory activity. So in these images, there are comparisons between the retinal scans when TIC-SNP is present versus when it's not. And the craters on the images, as he zooms in, are the dying cells, and the presence of BEST1 NRF2 with TIC-SNP stops and slows down the dying of the cells, as shown in the both the image he's circling and the one directly below it, you can see that there are a lot less dark spots and also known as dying cells. So then B shows how there was a 14% increase in a cell count with best one NRF2 when in comparison to when the control and best one NRF2 was.
No. I think that was my fault, Brooke, because I said that wrong. Remember, and then I corrected myself. Look, there's obviously a significant difference between the control and actually this best one thing is also a control. But there's a difference between these two and these two, right? The 14% actually refers to the one we know works, which actually Marco mentioned earlier, the CS, I don't know if you want to call it a mutant, it's really call it a variant, the CS variant versus the CS variant with the best. What is the best one again?
TXNIP C247S.
No. What is best?
Oh, what is the best one?
Yes.
The best one is a promoter for genes only in retinal pigment.
Okay, so stop for a moment, and I'm gonna do this. Everybody, what does that mean? What does that mean?
Is the cell count greater in the TXNIP CS best? The one all the way that has like the 10, has like the 14 above it. So that has the greatest one.
I'm wondering. Why did they use something called best one? What did she just say best one was?
Best one was a promoter for genes only in retinal pigment.
Okay.
Was a what?
Best one is a promoter for gene expression within retinal cells, period. So that means that whatever's attached to it, whatever gene you put under the control of that promoter, will only be expressed in retinal cells. You can do that kind of with any target you want. Like, name a tissue.
Muscle.
Okay. So think of a gene that is predominantly or only made in muscle. Anybody have an idea from honors biology?
Testosterone. Adrenaline.
Testosterone.
Hemoglobin.
What'd you say?
Hemoglobin.
No, you'll find hemoglobin in blood cells, so it is a connective tissue thing, but there is a protein. And those of you who aren't
myostatin.
You almost said it. Myo's the first part of the word. So
Myosin.
So myosin, especially MyoD, it's called, right? You only find that in muscle cells. So let's say that you want to label some muscle cells. You can take the MyoD promoter and put whatever it is that you want on the under its control, and it'll only, like GFP, right? It's fluoride, and that's actually what you're seeing right here. That's GFP, right? So you'll only see GFP in our example in muscle because it's under the control of a muscle specific protein. So here Brooke is talking about this best one, which is the promoter for something that is only in retina. And now what's NRF2?
NRF2 is a gene with the antioxidant damage and anti-inflammatory activity.
Okay. So if I'm only expressing that in cells that are retina cells, do you guys understand how useful that is? Like, you can target that specific tissue. So now go back go back to this figure here, comparing this and this. So this just has the TXNIP. All right, let me again. This journal club tends to be about methodology. What does this mean? You should all know, and otherwise you're all going to get below a 3 on the AP exam. C247S. C247S. You have seen this before. Now, maybe not that number, but where you have a letter, then a number, then another letter.
Statistics.
No, but thank you for trying. I do appreciate it. Okay, name some things that start with C.
Carbon.
Okay, carbon's a good guess. What else?
Cellulose.
Good. Cellulose is a good
Chromosomes.
guess.
Chromosomes.
Chromosomes a good guess. Okay, keep going.
Chlorophyll.
Nope. Good guess, but sure. But no. Now you're kind of getting further away. It's going to be the ingredient of something, right? What do genes code for? Oh my God, you should all be yelling. Yeah, thank you. You should all be yelling that. Genes code for proteins. If genes code for proteins, and proteins are made of what?
Amino acids.
Then what is C?
Cytosine?
That's the best guess so far. So cytosine is DNA. Same idea, though. So C stands for cytosine in DNA. What's C stand for in amino acids?
Codons.
No.
Is C an amino acid? Like
it is an amino acid. Which amino acid is it? Somebody's going to Google it at some point and end the torture. What is C? And then somebody else should be like, wait a minute. If we're looking up C, what is C?
Cysteine?
Cysteine is correct. Okay. Cysteine is the amino acid before the variant. Okay. 247 simply refers to the position in the protein, right? The protein starts with what amino acid?
Cytosine.
No. What amino acid is always the first amino acid? I'm terrible.
Methionine.
Methionine, yes. So methionine is in position 1, and then you keep going and you read the next one. That's position 2, then position 3. Well, in position 247, it used to be a cysteine, and now it's been converted through a mutation into S, whatever that is. What is it?
Serine?
Serine. Yes. So a cysteine to a serine, and if you know the structure, which you don't, but when you take biochem in college, you'll know the structure. You'll have to know all of the structures of the amino acids. You know that cysteine contains a sulfur group, which is really important for holding the three-dimensional structure of a protein together. But if you replace it with something innocuous like serine, the structure of that protein kind of falls apart. It doesn't hold itself up so that it's functional, because sequence equals structure equals function. Okay, so what this is saying is TXNIP with this particular mutation or variant is actually, it helps to rescue the phenotype. It actually helps stop the retinitis pigmentosa. But they're like, but we can make it better. If you put this NRF2, which is what again, Brooke?
NRF2 is an antioxidant is a gene with antioxidant damage and inflammatory activity.
Anti-inflammatory activity.
Anti-inflammatory.
It reverses oxidative damage and does anti-inflammatory. So and it's only turned on in best one producing cells, which are retinal cells. So you have a combination of this really good variant of TXNIP and something that's anti-inflammatory, and you actually see it gets even better. So what this is doing, well, I'll let one of you guys or Brooke kind of take over from there. So what's the
is best one naturally occurring?
That's a good question. Brooke, do you know? I don't think you would, but
I'm gonna go with yes.
it is correct. Yes, it is naturally occurring. Yeah. I'm gonna get rid of me. EOS out. Okay.
Okay, so then as we continue on this figure 8, we then go down to D, which it talks about DGFB1 or beta 1. So it's showing how DGF beta 1 is similar to BES 1 and NRFD2, in which it stops / slows down the death of the cells when it's present with TXNIP. And then we'll go to the next slide, which is conclusions and implications. So ultimately, the paper's conclusion stated that TXNIP prolongs the RP cone survival and visual activity. So my conclusion is I believe that the information in the paper was expressed well. However, there was bad figure legends and overall needs cleaning up. So then for implications, I said that this particular study could be replicated for humans in which TXNIP could be administered through injection as it was for the mice or rats, and it could be tested for similar retinal degenerative diseases in further studies. And that's all I have.
Hold on, I want to just bring us back to this, and I'm gonna move you. Okay, there we go. So, with the results we saw, what does that have to do with this conclusion figure?
Are you asking me?
I'm asking kind of anybody.
We're basically saying that TXNIP is inhibiting glycolysis and it's improving the mitochondrial health, as well as is contributing to lactate catabolism.
Okay. And just to clarify, what is catabolism?
Catabolism is the breakdown of the lactic acid.
Right. Which is the opposite of what word?
Anabolism.
That's right. Anabolism, right. And so and the way I remember that, listen, I get that confused all the time. The way I remember it is that the anabolic steroids build you up, vitamin S, right? Builds you up, and catabolism must be the opposite. So breaking you down, turning you into goo. Should this have been published in eLife?
So I don't necessarily know about eLife because, as I said earlier, it does need some cleaning up in terms of like the figure legends and how the information is presented on the actual paper. I do believe that the study they're presenting is a good study and that they did have good figures that they used, but overall it just needs cleaning up and better explanations of what those figures were.
Okay.
I have a question. How do people use TXNIP? Like, do they like?
I'm glad you asked that question.
Yeah.
So it's actually placed into like the retina through injection. So what happened was the TXNIP was injected into the retinas of the mice so that it could be used to prevent the degeneration of the glucose.
But methodologically, how is that done?
What do you mean?
I think he's asking, like, how do you get it in there?
Like, do you just inject like a needle into their eye or something like that?
Yes.
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 It's gross. Yeah. And they're testing it in humans. The humans are the whole bunch of macular degeneration and a bunch of other disorders are being tested this way, where they are injecting the eyeball with all sorts of things. I mean, you guys are of a generation where when you get to be my age, you will not be wearing glasses or contacts. You're just going to go you're going to go to the Walmart, put your face on a chin strap. They're going to scan your eyes and then shoot some lasers into your eyes to do LASIK surgery, and boom, you'll be done. And then you're going to live forever. That's the way it is. Yeah. Okay. Questions, comments, concerns? Oh, come on. We still got plenty of daylight. We're fine.
I have one more question about the paper.
Okay.
If you look through it, I'm not sure if there might, but I'm just wondering if there are like any side effects to it. Tixnip.
That's a great question.
I have no idea. I'm not gonna lie.
I think
because it can't be like all like 100% success rate, right?
Why not?
Does that happen a lot?
Not with broad spectrum general like whole body things, no. But what did they do to make it specific to the retina?
They used best one.
Yeah. When you make something only usable in the one tissue you're targeting, you're going to really reduce the number of side effects. For example, let's talk about antidepressants. Okay, as an example where you can't really use cocaine. If you were able to start, I don't know, knocking down serotonin transporter, but only in the brain, you would have fewer side effects than regular antidepressants, because regular antidepressants affect the transport of serotonin all over your body, and you use way more serotonin in your gut than you do in your head. So by making it more and more specific and timed better and better, we're going to reduce the number of side effects. Making those connections is kind of why we do this. I think that's kind of cool, right? And so like in the future, if you have to solve a problem of, I only want to know, I only want to see these neurons marked if they are this type of neuron. Now you know how to do it, right? You could just go find, like, let's say, here's another example. We said the myosin gene for muscle. What if you only wanted to see glia? You could use something called glial fibrillary acidic protein, which is GFAP. So if you only mark GFAP, then you're only going to get those glial cells, which is really powerful. That you can only, we've seen it also with Cree-Lox. Who's the one who did the Cree-Lox? I forget. One of you
I think
guys.
that might have been me. It sounds familiar.
Yes, it was you. It was you, right? Cree-Lox put the Cree under the control of whatever gene of interest you want, so you're only affecting the one type of cell, right? Like there are genes where if you knock it out through the whole body, you die. But what if you only knock it out in a certain type of cell, right? Now you can really study what's the effect of knocking it out in that one type of cell. It's really powerful, and that's what they did. And they're using it therapeutically, which is even more impressive. So should it be in eLife? Your final say, what, Brooke?
Not necessarily until it's fully refined and better explained.
Okay. Anybody else have any points? I think we were good in that. Thank you, Marco, and Johan for asking questions and stuff. But and for you, Marco, for you actually trying to do figure 2 or whatever it was 5. That's what Journal Club is. And when we're in person again next year, that's what it's going to be. The person who is responsible for it gets to choose it, introduces it, usually does the first figure, and then they only come back to conclude. And everybody else is responsible for having read the paper and actually covering those figures. That's what this should be. It's a little more pressure, but it puts you on the spot so you learn stuff. Other comments? You tortured enough? Okay. Well, I will see you all tomorrow. Would Miss Liff and Miss Hughes stay on?
Have a good one.
Thank you.
Good job.
Thank you.
Thank you.
Thank you.
Thank you.